Okay.
Um,
Well, welcome to the Cure Lumpy One podcast.
Thanks so much, Chris. Thanks for having me.
Okay, so we have today Richard Novak of Unravel Biosciences and Shift. So, Richard, we're really, really excited to talk to you. You're helping us set up this YouTube channel, and we really think that obviously your service and your business is incredibly interesting to parents in our position for parent-led research, which is exactly where we find ourselves of parents with an ultra rare for our daughter Aria, who has an LONP1 mutation, and she's the only person in the world with this mutation. So yeah, we wanted to have you on because we think that it's incredibly interesting what you're providing us, the potential right now that we're in our fundraise at curelomp1.com. So, and your team is part of what we call the A team. We have people from all over the world that we're trying to collect, and we're very proud to have association with such bright minds. And really, we just wanted to bring you on. So we're still learning about Unravel and also Redshift, and it's an opportunity for you to explain to people in our shoes, tell us about the firm, tell us about you guys and why you were set up in the first place, and the sort of the genesis of the company.
Yeah, thanks, Chris. Well, it's wonderful working with you and Andrea and see what all you're doing for Aria. It's really heartwarming. It's something that we always see with families. Unfortunately, rare disease families have to become the scientists, the drug developers, the experts, especially in a case like yours in Aria's, where there's very little other data to support any understanding of a given disease. And that was sort of the motivating factor for Unravel, for how we started it in the first place. So my co-founder, Frederick, and I, we were both at the Wyss Institute at Harvard, and it's a very translational institute. We were both on the staff side. So the non-academic side, really helping commercialize technologies, drive programs out of the lab, actually. So the mission is really, you can't have an impact unless the technology leaves the lab. And that resonated beautifully with me. So I had a background really bringing science education and engineering education to developing regions, and developing some other solutions that were more led by those experiencing the problems. Like, I will never understand the problem, but I can collaborate and help drive some sort of positive impact. That's the hope, at least. And so that was really the motivating factor behind Unravel. It came about because we were working on some very challenging disorders and disease states. And they were just extremely tough to approach with the traditional biopharma mindset. Typically, that means start with the target, do some preclinical development, and go to the clinic. And that linear sort of reductionist approach is really ineffective. And if you look at the statistics in clinical trials, that backs me up. Very ineffective in actually the very complex disorders. And certainly LONP1 and a lot of these mitochondrial disorders are absolutely in that category. And right. So what we did was really come up with a novel way of thinking about biology, right? Fundamentally, we don't understand most of it. And just let's get that ego out of the way. We don't know.
That makes me feel better, because this made me feel very thick and suddenly having to school up on some of this stuff. But if you're saying that, then that really confirms the problem, right? It kind of just, even when you start, if people really understood what was happening inside the biology, the bioengineering that's happening, it just looks like somebody's come up with some crazy ideas. And if somebody told you about it and described what was happening in cells, it sounds like unbelievable. And when you actually see some of this modeled, and actually the cell behavior and mechanisms and machines that are happening inside the cell. So, yeah.
Absolutely. So it's stunningly complex. I mean, every day there are thousands of papers that come out trying to, they show some sort of new mechanism, new target, new activity that wasn't before uncovered. And that's the fantastic, interesting side of basic science research. Now, the challenge is, if you have a patient with a rare disease, and we work oftentimes with the most challenging, most severe cases where you typically don't have 20 or 30 years to understand a disease, and yet the patient needs help. What do you do? And that was really what gave rise to Unravel, which is we found these technologies, developed these technologies at Harvard that enabled us to think about biology in a very different way. essentially simulate what actually happens in a patient without having to understand it from a mechanistic standpoint, but in a way that's strong enough to let us actually see how do we mess with it, how do we perturb it, and then it obviously guide it to a way that's favorable, that restores health, right? So in that, what I just said, none of that involves knowing a target, knowing a disease causing mechanism. um, you really want to work from the patient, and then work your way backwards towards a drug, and then at some point you can work towards a target. And essentially what we've done with Unravel, and that's where the name comes from, is really unravel the disease from a patient to a therapy, and then we can kind of fill in the basic biology if needed. It may not even be needed, and so it's really a very different way. And it runs very much contrary to how drugs are developed today by big pharma. And we found that we don't have $1 billion to throw at a drug with a 95% chance of clinical failure. We have to think about it smarter, and that was really the rise of Unravel Biosciences. Now, what did in the process, we started working on rare diseases. Started with Rett syndrome, and that's actually headed to 2 clinical trials. We just got orphan drug designation for the drug that we uncovered using our platform. And there's investor interest around that. So we started Unravel, right? So we spun out of Harvard about 3 years ago now with this extremely powerful platform. And so the whole idea was, how do we advance a therapeutic? Basically, what our platform does, it has a deep understanding of RNA biology, of how the genes actually interact, not from a mechanistic standpoint, but if you perturb gene 1, how does it affect the other 25,000 genes? Just a correlation model. You don't have to know how it actually physically works. But if you, let's say, the equivalent is a great example in traffic maps. So we like to say we have a Google Maps for health. The whole idea is that you have a starting point and you have a destination, and you have to get through some amount of, let's say, a city in between. Traditional bioinformatics works by asking what's different. Well, knowing that you have a bed at home and not in your office doesn't help you find the way. And you run into these sort of red herrings in traditional bioinformatics. You have all these things that are different. Many of those don't actually help you answer the question, how do you actually fix the problem?
So you're referring to bioinformatics. Is that what you just said?
That's right. So that, just to define it, is really computational analysis of biological data. Typically, it's RNA data, DNA data, protein, all these different bits of information that you can collect from a patient or some other biological living system. The computational analysis of that, try to make sense of it, make it human interpretable, is called bioinformatics. Now, the challenge is that typically says, okay, you have a healthy state and you have a disease state. What are the differences? That's typically what most of the algorithms ask. There's this whole onslaught of AI and machine learning that gives you even more refined differences and finds the subtle differences between the two groups. What it doesn't do, it doesn't tell you how you actually travel between, let's say, a disease state back to a control state. And that's exactly what Unravel is.
I've heard you talking about being agnostic to certain, how that happened, or the mechanisms. That makes sense now. Now you're explaining it that way. So thank you.
Yeah.
So
and just like
so this is AI modeling. This is neural networks we're talking here.
Yeah, a form of it. So essentially, it is a network model, not specifically a neural network, but the concept is sort of similar. Everything talks to everything else, right? Just going back to this Google Maps analogy, if you have a car that breaks down in an intersection, obviously the cars around it will be immediately impacted, but over time that traffic jam will propagate into the neighborhood. So everything is impacted by everything else to some extent. And essentially what our machine learning does, it finds these probabilities, what's the fractional impact of this gene on this other gene, saying like, what if a car stopped at this intersection versus this other intersection? How would it impact the streets 2 neighborhoods away, right? There's some impact. It could be very little, it could be very large. You don't know until you start analyzing it. And so, for example, if a car breaks down on a main highway versus like a neighborhood cul-de-sac, 2 very big different impacts, right? And so that's what essentially our machine learning approach studies, is really understanding those connections, even without those connections having been necessarily identified in the scientific literature. So unlike a lot of machine learning companies, we don't mind the literature because it tends to be biased based on what people were, one, interested in, and two, got funding to study, right? Now you come in with a new gene,
Amen to that.
they
Amen
never
to
have been
that.
studied.
Amen to that. That's a massive problem in science and medicine, as far as I'm concerned. But yeah,
Yeah,
so
yeah.
I w is there another way of sort of deeper dive on kind of how that works when it comes to, so for example, what we want to do is we want to be able to work with you, and that's what our fundraising is in part, to bring you into this team because we're so excited about working with you and the Shift ability to make that sort of accessible to us. To what's the end goal here? The end goal is looking at something that's therapeutic for ARIA in some way. For our story, Aria is the only surviving person with this mutation. Similar mutations in children in the record died at 20 months. And so we just don't know how much time we have with our angel. And so you guys turning up and saying, we understand that and we think we can do something. And obviously the power of kind of, I'm guessing, brute force, if you can just throw CPU time or something at the problem. I don't know what the limitations are for how that works. Um, but for, explain to people, I think it would be really interesting to understand, how does this compare? Like the old model might have been drug repurposing focused on yeast models and that type of thing. Is that true to say that?
Yeah, so
And
that falls in the category of drug development or repurposing that's called phenotypic screening. I mean, the name aside is
okay,
basically you have a living system, you make a model of your disease in that living system, and then you can basically throw molecules at it and see which molecule helps in some way, right? And you have some way of
right.
reading it out. In yeast, for mitochondrial disorders and other related disorders, that has been fairly
Mm,
successful because they have mitochondria just like humans. The
hmm
energy powerhouse of the cell is pretty conserved across organisms. So they actually
hmm
are quite a good model system in that context. And
Yeah,
then you have to screen thousands of molecules in actual physical dishes in a laboratory, use robotics, and it might take you a few weeks, few months to screen a few thousand molecules. Essentially, well, but the upside of it, you are working with a real living system, no
yeah,
question, right? Even the
yeah,
parts you don't understand are in that system, right? So that's the upside. Now, it's not a vertebrate, it's not ARIA. So you always have to deal with the fact that it's a
yeah.
model system versus an actual patient. You obviously can't
We
dose
understand even,
8,000
we understand
molecules.
even I think we've got a delay on the line, but forgive me for cutting across. Even with critically on the LONP1 in a yeast model, there is some differences, I think, in the in the yeast expression of the LONP1 versus human expression.
That's right. That's right. There's some differences. Some differences are known, some are unknown, right? And
Yeah.
so you get back into the question of how much biology do we understand? Is this the correct model or a correct model, a valid model, given the context? You don't know until you go from that model to a patient, and
Mm.
then the reality check comes in.
I guess I'm interested how you calibrated in the first place. How do you calibrate those tools, or your platform as you refer to it? I don't know if it's given a name
Yeah.
or a secret name or a name that you refer to your platform as. But how was that calibrated against things like yeast or real organisms?
So it's a human model. So the platform is called BioNav. So that's really the computational, drug and target discovery platform. Essentially,
Hmm
what it does, the input is RNA sequencing data. So looking at how the genes are transcribed, going from DNA to RNA, and that's where one of the key parts of living cells, living organisms. And then we take that information. We have an algorithm that converts it from this list of genes and how much of each gene is in the tissue being collected, and converts it into essentially this Google Maps traffic map. Where are the traffic jams? What's going on with that patient? And we compare the patient data relative to a healthy,
A
usually first
control. So
degree
in
means
this
of.
case, you would sample Aria and you would sample Aria's mum.
That's right. That's right. That's typically a good scenario, or oftentimes it's the parents. Sometimes they're siblings of the same sex, and actually that
Ah,
matters quite a bit. The idea is that we're trying to see, okay, how do we make the patient as close to whatever that healthy
mm,
control, first degree relative. So a lot of the genes are the same, a lot of the environmental factors are the same time zone, typical similar sleep schedules, diets. all of that plays into health and how people
Mm.
respond to drugs, the response to genetic mutations, other factors all have an impact. And the idea is that by looking at the RNA, we capture all of that. So it goes beyond DNA analysis. So DNA is typically how patients get diagnosed because there's a mutation. You can do DNA analysis. It's actually very robust because DNA doesn't change, except, I guess, in cancers. But in the case of these genetic disorders, the DNA is not changing, but the disease is, right? And
Okay.
so it's not because of the DNA alone. The DNA is encoding certain genes and has certain predispositions, but then it has to interact with environment. The in this case, ARIA is growing, developing. That all factors into how the disease progresses, doesn't or doesn't progress,
And the
in
bit where
response to
you could
medication and other factors.
the bit where you could help me out was just understand from a technical point of view how there's an RNA is produced from the LOMP1. Are we focused on the whole system? And I'm guessing all the genes that are being expressed in those samples. So that would mean there's lots of RNA, right? And I guess I'm just curious about how much RNA and how you look at all of the RNA together.
Right. So, from a diagnostic standpoint, what ARIA received as the diagnosis was that LONP1 is mutated, right? And that obviously is the root cause of this disorder. That part was clear from the diagnosis. It does not tell you how to treat it.
Yeah.
And so that's sort of where we come in, and it doesn't even tell you necessarily how the disease will progress or other aspects. So there's this type of analysis that oftentimes a lot of foundations do, which is called genotype phenotype analysis. So genotype is the DNA. Where is
Mm.
this gene mutated? Let's say where's LONP1 mutated for
Hmm.
each individual patient, and then
Hmm.
correlating that with symptoms. Let's say seizure, developmental delays, cognitive function, etc. That relationship can be correlated, right? Now, sometimes that's quite promising, but many times there are a lot of sort of confusing results. We sometimes actually now we work with one foundation now. There are two girls from completely different families that have identical mutations in the particular gene of interest. Are on the complete opposite spectrum of the severity of the disease, right? Clearly that gene alone does not explain what is actually happening in those
Yeah.
patients. It's
Yeah.
absolutely the gene causing the problem. That's not the question. The question is
Yeah.
what else is involved. And essentially, what the RNA does, you mentioned there's a lot of genes. There's about 25,000 genes in the human genome, give or take. It turns out they all have these variations. There are these inherited factors. There's spontaneous
Mm.
variations, mutations that are not causing disease, but they can certainly affect the outcome of drug treatment, the disease itself, the root cause of the disease, and a bunch of
Hmm.
other. It could be, how quickly you metabolize a drug. I mean, that's been studied very, very deeply because it impacts
Yeah.
every drug you take, how much you metabolize it. There are also some mutations that can actually help counteract the disease in a patient. This has been studied, for example, in Parkinson's and Alzheimer's patients,
Hmm.
where you might have two siblings, even fraternal twins or somebody very, very closely
Hmm.
genetically related. One develops the disease, let's say, 30 years before the other. What's going on?
Wow.
It's not this. It's not the what you think is the causative gene. I mean, that may be causing the disease, but that's not the
Hmm.
full story. And essentially, that's where
Yeah.
Unravel's BioNav platform comes in. It's not about that one intersection of the car being broken down. It's the
Hmm.
whole city map that you care about, and how do you actually travel from a disease state back to a healthy state. And essentially,
Hmm.
that's what our machine learning platform has identified. Now, when it comes back to patients like Aria, traditional AI approaches require tremendous amounts of data, right? I mean, just staggering
Okay.
amounts of data. What we've done that's quite unique is we've essentially, you asked how we sort of calibrated our model. Well, so our model is actually human data that's driving it entirely. So we can feed in data from other species, but everything is processed in the context of a human. And so
Sure.
we've looked at the 25,000 or so human genes, and we've analyzed if you perturb one gene, what effect does it have on all the other genes? And we
Yeah.
obviously went through that iteratively, and then that's how we built up the model. Now that took a massive amount of computation, but it's done.
Wow.
The whole idea is right that we looked at um, all this staggering amount of data from human tissues and where essentially one gene was perturbed, and we looked at what does it do to all the other 25,000 genes. And we went through that, and this is where we built up this very deep machine learned model of human health, right? And that took a tremendous amount of data processing. Now, what that means is that, and we did the same thing for about 40,000 existing molecules. Instead of a single gene perturbation, now add aspirin, add Tylenol, add a statin, and ask no other information or no other question other than what effect is it having in this network state. So now we have this really interesting scenario where we don't need a huge amount of patient data. We can start with a single patient and a single matched control, and we can start asking the question, how do we convert that patient into the healthy control? And essentially what we do, we run simulations, we import patient data, which actually I can talk about in a moment, but we import patient RNA sequencing data into our platform, and then we can essentially simulate what happens if you give someone aspirin? What happens if you give someone this particular statin? And then we can see where does that model tell us the patient would go. Most drugs are irrelevant; they just don't do anything, as we know. Also, clinically, that's true. Some drugs may make the disease even worse, right? They may be something you don't want to take. Other molecules, and these are the ones that we're looking for, are which ones actually go from, let's say, Aria's state that we have interpreted to where her mother is, right? So the idea is to drive that path from a disease state to a healthy state. And essentially we run, contrasting that with the phenotypic screening that we discussed earlier, we can run 40,000 drugs in a few minutes on each patient sample. And so we can even look at drugs over time, drugs at night, drugs during the day, which ones could actually be therapeutic in that patient's particular context. Exactly. So we have a human model of health. We have a database of about 40,000 molecules, including a lot of the repurposable approved molecules for some indication. And then we can essentially go to just individual patients. And really, we are very much patient-led, patient-centric, because we see every patient as an individual patient or disease, independent of how they were diagnosed. And so what we do is we essentially import the patient's RNA profile into our database or into our BioNav platform. And then we essentially run, if you think of it as, a N of one or single patient clinical trial on 40,000 molecules, one at a time. And let's just see in the computer what happens. That's all we do, right? I mean, that's all we do. It's a complex process behind the scenes. But it's this very simple question that we're asking, right? Which molecules help the patient, which ones are neutral, and which ones might actually make the disease worse.
So I could visualize it. I could visualize it like some kind of like a graph or some kind of state of this is dysfunction, this is disease, and this is the control, this is health. And you're, in my layman's terms, would be understanding, does this molecule move us closer or further away from that? Is that so? I'm guessing
that's right.
You're doing something.
That's right.
Okay, and so from an RNA point of view, are we looking at only the RNA that comes from the LOMP1, or are we looking at all of the RNA that would be expressed in is it a nasal swab or a cheek swab?
Yeah, so let me talk about that for a second. So, of course, when you have a computational platform, the patient's not digital, so you have to somehow digitize them or incorporate them into the platform. And so the way we found out how we can work is through nasal swabs. So thanks to this lovely COVID pandemic, everybody knows how to swab. And we actually found a way to, instead of looking at the pathogen side, the virus or bacterial side of the equation, when you do a nasal swab, you scrape up some of your own cells, the human cells. What we were able to do is show that from just the few cells that you scrape up, and it's very complex inside your nose, your blood vessels close to the surface, immune cells, epithelial cells, sort of like skin, but sort of almost like inside of your lung is actually what your inside of your nose looks like. We basically take all that information, collect the cells, and convert it into RNA data, so-called RNA sequencing. And this really builds on the Personal Genome Project over the last 25, 30 years. all the technologies that were built up, including the ones that were used oftentimes for the DNA diagnosis that Arya received, for example. But instead of the DNA, we look at the RNA. But to your point, it's not just LONP1. This is completely, as you mentioned, the term of agnostic. We don't know what to look for, so let's look at everything. And that's really, we don't know what we don't know. And that's really the guiding
Yeah, yeah,
philosophy.
yeah,
The
yeah,
only
yeah.
thing we know is that the disease is in that patient, and that patient's disease
Mm,
is different from
mm,
potentially a different patient's. We make no
mm,
assumptions about any of that.
mm. It sort of seems to really embrace the complexity and remove assumptions as a model,
Absolutely.
and
These
that's what,
diseases
to me,
are
that sounds.
staggeringly complex. And so we don't know what we don't know. So let's just take as much data as we can and learn from it. Have the data tell us what we should be looking at.
So the specific thing about the LOMP1 expression and the RNA versus all the RNA that might be expressed in those cells, help me on that.
Yeah, so LONP1 is the cause of the disease, but what we're interested in is how to fix it. So it's a slightly different question than a diagnostic question. So we're not on a diagnostic odyssey, we're on a therapeutic odyssey. What's very unique about Unravel is that we make the hypothesis that how you treat a disease does not require reversing the disease itself, how it was caused. There may be other mechanisms, and there's a lot of data out there in many other diseases where that's true. So it gives you a lot of flexibility on how you could potentially treat the disease. Biological systems are extremely so-called plastic. So if you have some sort of stimulus, whether an injury or something, extra nutrients, the body adapts. It doesn't need to be this very consistent system, and this is because of evolution. The organisms that were able to adapt survived, right? That's really the
Yeah,
nature of it. So
yeah.
humans, as an organism, are very able to adapt, including at the cell level. So what we do is see, okay, the mutation in LONP1, in Arya's case, causes the disease. Let us have the software model. How do we fix it? Now, one mechanism could be by focusing on something directly linked to LONP1. In this case, clearing out bad proteins in mitochondria. That's sort of the function of LONP1. Maybe it's somehow disrupted. It might have other functions that have never before been documented,
Yeah.
right? For all we know, that
Yeah.
happens all the time. Our platform lets us work with that sort of uncertainty or unknown and just simulate. Okay, if you add a drug, right, what does it do? Does it fix things? Again, from this
Hmm.
biological network standpoint. So we look at all the,
And how are you measuring? How are you measuring the fix, as it were? What is the? Because you're not looking at the RNA at that point. You're looking at something else, right? That says
so we still look
so
at the
like
RNA.
the outcome. Okay.
Yeah, so we still look at the RNA, essentially sort of think of it as a network state, right? Like a pattern that you're going after. So there's in this case Arya's mother's RNA pattern, and that we're defining as healthy. This is where we're trying to get to. And then
Yeah.
there's
Yeah.
Arya's pattern. Overall, it'll be very similar, but there can be some areas where it's not the
Mhm.
same.
So
And so
it
we
could. You are you saying it's ultra focused on simply the RNA, and that's essentially what we would assume if we get to correcting the RNA expression? Is that what we're saying?
Right. So the even though we're reading out only the RNA, again, it's a biological system. So DNA gives rise to RNA, RNA gives rise to proteins. They're all these metabolites, modifications. It's a very complex. It's not a nice line that we might have
Hmm.
learned early in school. Everything's talking to everything else. But by using RNA, it's a very robust way of collecting information on this whole complex system. Yes, you absolutely have gaps. We are not reading out the protein. We're not reading out metabolites. We're not reading out modifications to the proteins, a bunch of other factors.
Yeah.
But at
Yeah.
some point, it feeds back into the RNA. And essentially, that's sort of like with Google Maps. You don't have to track every single car on the highway to have a sense of where is there a traffic jam. If you
Okay.
track every 50th car, you probably have a pretty good idea, right? And it's the same thing
Yeah.
with
Yeah.
RNA. It's obviously imperfect, but it's good enough to let us see what's going on at this really complex system level. And that's really how we work. And so it's all the genes. They come from the swab. To our surprise, that actually gives us really good correlation with what's happening in the brain and other tissues. Um, and we've been able to use this for other patients where basically the top drug that we identify, essentially this in silico computational clinical trial that we run, we took basically the drug that was computationally predicted to be most helpful. There were a couple families that started taking it for two different diseases and have had some really profound positive effects, right? So the idea of
It's
repurposing can
incredible.
be actually very, very therapeutic. It's not sort of just historically, it's been focused on mitigating symptoms. So, for example, if you have seizures, how can you prevent seizures? If you have, GI problems or cognitive problems, let's put that aside and let's do something else later. That's historically how repurposing was thought of, or even just drug development. Let's come up with
Yeah,
a better
yeah,
seizure medication. What we
yeah.
do, thanks to looking at it from a network standpoint, the hypothesis, which so far has been panning out quite nicely, even in patients, is if you can fix that RNA network, right, the one that we've been talking about, this kind of computational network of the
Yeah.
patient's disease state and revert it to that of a control, you fix a
Hmm.
lot of problems at once. So essentially, it's counteracting sort of that the cause of the disease or the impact of the disease in some unknown way, right? Or sometimes known, sometimes unknown.
Yeah, I
That's
hear you.
really how it approaches
I'm
it.
still trying to get my head around it, even though, but it sounds my kind of way of approaching problems, and I think just incredible how people have figured this out. But I think it sounds, you've got to harness nature, right? You've got to say you've got to respect it and remove all the assumptions. And so for me, working and solving that problem in that way is working with that elegance
Mhm.
and that beauty and accepting that we there's so much that we don't know. But I even said, even though you've been working with me so well trying to explain it, it's still kind of hard for me to understand, and I wonder for other viewers as well for them to take away. But I think for parents in our shoes, um, give them. It gives people hope. It gives people tremendous hope. It's definitely light in the darkness for us and our family, that we're doing this fundraise, and we kind of implore people to sort of check out Unravel and to check out our website to curelomp1.com and hopefully donate. And if you can't donate, share. And so that we kind of want people to be aware of what you're doing, Unravel Biosciences and also Rare Shift. So probably give people a little taste on what's the difference between the two, what Rare Shift is versus Unravel Biosciences, and also we've talked about in silico, which is super interesting. What about in vivo, an animal study to move things forwards and help people in our shoes, parents who might be out there looking for help? How should they understand and interpret this?
Yeah, thanks, Chris. So, really, we have sort of two arms. We have a very innovative business model. And the whole idea is that we have this very powerful platform that we can use internally as a biotech company, which is what we are. We're a therapeutics company based in Boston, sort of one of the major biotech hubs. And our goal is to develop drugs, right, and eventually sell them and develop them through the clinic and into the market. So that's really what we are as Unravel. Now, what has become very apparent, there's this massive unmet need in neurodevelopmental or just genetic disorders, um, usually neurodevelopmental because that's been sort of the biggest gap historically. But families started coming to us essentially wanting repurposed drugs because they heard that we use existing molecules for discovering new mechanisms, new targets, right? And so
Yeah,
for us, what's a discovery tool is potentially either a temporary or potentially the end treatment for an actual patient. And so over the last year, a year and a half, we actually started partnering with families, letting them use through a nonprofit service model, our platform. And so our platform consists of this in silico model of human health, which is absolutely revolutionary. And where in a matter of a month or two, we can get to a list of existing molecules that you can work with your clinician to take off label. We cannot encourage that. That's entirely a personal decision. But we can certainly generate the data for that. But then from a longer term standpoint, we can go all the way to clinical trials, either with existing drugs or use that information and develop newer, potentially even better molecules that are now tailor made for the therapies. And this really addresses a major unmet need in the rare disease space, because if you have, let's say, one patient, no pharma will touch that. That's personalized medicine is not a good business model because you only have
No,
one.
exactly. And this is the fundamental problem for people in our shoes, and this is why
Exactly.
we're completely alone without people like you guys.
And so what we did very intentionally was open up this platform just through a nonprofit model. So it's not free, but it's just the bare minimum cost to operate it. And families and foundations get repurposing, and we get data and the potential path for commercial development, right? And so it's sort of a win-win where we charge the minimum, but then we can be your partners in drug development for both existing drugs. So this could be your
Hmm.
traditional repurposing path. We're also doing this, making new formulations, synthesizing drugs if they're not readily available as generics. Case by case, we can do all kinds of things. Our chief medical officer has brought 8 drugs to market. So, as a company, we've done this many times. But also being the partner all the way through new drug development, kind of building on the data, including clinical data, oftentimes showing, hey, this path will work. This target that we've uncovered, that our computation tells us is really, really interesting therapeutically, this will work. Let's build a better molecule. In one case, we're doing this for Rett syndrome. We're using an existing oncology drug, but we found a new target, and it turns out it's metabolite, right? So it goes through your liver, gets degraded, but actually that degradation makes an even better molecule for Rett syndrome. And so we're actually commercializing that as a new molecule. That will, of course, take years of development. But in the meantime, we're running 2 clinical trials to show that this existing molecule could be, one, an immediate treatment,
Hmm.
and 2, showing that this mechanism that we've identified actually works in patients, right? So
How do you
then,
think that could work for Aria in that sense? What do you predict? Once we could hopefully get this going, what would that look like? Do you think for Aria, given what you know about LOMP1, or um, this is useful to know personally. I'm kind of intrigued.
yeah, so really we get this list of drugs out, right? So these are basically the personalized list of drugs for ARIA. And we always say that the disease discovery and therapeutic discovery has to happen at the individual patient level. There is no average magic patient. It is
Hmm.
ARIA, it is Billy, it is whoever, right?
Yeah, yeah.
They have their unique combination of genes, environment, and even just the progression in time for their disease. That makes the disease very unique. Now, we basically take it on ourselves as Unravel is to figure out, okay, we can do discovery at a personalized level. How do we actually make this make commercial sense? And that's really the transition from rare shift, which is patient by patient nonprofit model, to
Mm,
there may be a business opportunity here, right? And if there is, everybody wins. It's the idea is that feeds back into the patients as well. But it is up to us to essentially find connections across these rare, ultra rare, and even N of 1 disorders and form basically commercial opportunities. And that's an
mm,
Unravel
mm.
problem. But we're basically trying to make sure that at least one thing that you guys as parents don't have to deal with is the business side of things. You're
Yeah, for
already
sure.
scientists, you're already caregivers, you have all these other responsibilities. Let's not add, biotech executive to that as well. And so the whole idea is that Unravel sort of serves as bridge between, to some extent, kind of a nonprofit early discovery, oftentimes, aligned with academic discovery, but then seamlessly transitions into, biopharma and including 505(b)(2) reformulation, at least with the US FDA, but all the way to new drug development. Sometimes you have to develop a new molecule. An existing molecule may
Yeah,
not
yeah.
accomplish what you want. So to answer your question, we don't know, but we will find
Mm.
out relatively fast what that looks like, right? And so
What kind of timelines would you be thinking?
So typically, it takes about a couple of months, unless there's a urgent need to accelerate things to work with the patients, get their RNA sequence run through a platform. Now, you also alluded to the fact that we also work with preclinical models. So we actually use tadpoles, so your Xenopus laevis, African clawed frog embryos. We genetically engineer them with, let's say in this case, LONP1, to make little patient avatars of whoever it is, of their particular disorders, to be one or more genes. And
Hmm.
then we can actually screen the molecule. So that's typically how we progress. So we go from patient to a drug list, look at the drugs, look at the ones that make the most sense for
Hmm.
translating,
Hmm.
go back into these tadpole models, and take us a couple of weeks to make a tadpole model. So it's actually
Hmm.
really, really fast, much faster than mice, even faster than like even fruit flies, and then lets us work very quickly. But importantly, this is why we like whole vertebrate animals, because we can ask about neuromuscular function, seizures, growth and development, sleep.
Right.
They sleep at night like
Right.
humans do, right? All these factors, even some social interactions, autism phenotypes, we see that, and we have this wonderful computer vision algorithm that we developed. We basically, through a camera, we can analyze all these metrics for thousands of animals at once, right? And so
Wow.
that lets us actually essentially take Arya, which is one patient, and you want to be, of course, very careful, safety first in terms of anything that actually ends up in ARIA. But this is where you go into preclinical models. Use the animal
Mm-hmm.
model,
Mm-hmm.
test all kinds of hypotheses, all kinds of drugs,
Mm-hmm.
drug combinations, do the target discovery, and we can drive that very quickly. So in a matter of months, we can get to, let's say, a therapeutic mechanism, have a molecule that can engage it. Ideally, it's a molecule that can be consumed by a patient already, so it's already
Yeah.
approved, and
Mm-hmm.
then we can move towards a clinical trial. So we've done this now several times, where in always under a year, but now as low as about 2 to 3 months, we can go from working with a patient, developing a tadpole model, and showing that potentially, if the patient and the family decides with their clinician to do something off label, we can get clinical, at least proof of concept, very quickly.
Okay.
We can
Mm-hmm.
do very rapid proof of concept clinical trials under the appropriate regulatory guidance.
Mm-hmm. Mm-hmm.
But then we can also show that it works in these tadpole models. Oftentimes, in
Mm-hmm.
parallel to developing mice or other more
Yeah, I
established
was thinking that in Aria's
programs.
case, would we then look at mouse models too, and
Yeah,
this type of thing? No, that's really, really interesting, and I appreciate you running through that. It's the gene itself, in terms of taking it, you know to a state where it can impact other people. And certainly, we hope that when people are donating to help us do this work, we're looking primarily to extend Aria's life so that we can, um, work on additional longer term treatments for ARIA. But the gene in question, LOMP1, increasingly is of interest from, as I understand it, things such as cancers, and you will know more than me clearly on its impact. But if you were to develop a therapeutic and took it to that point where you could, produce something on a scale that has an impact on, who knows, things like CODAS syndrome, or potentially cancer treatments or something. It's difficult to predict, isn't it? But it's certainly an endeavor that we see across the other partners that we're working with, and we're so pleased that people are taking an interest, because of that. But to me, it seems that there could be, a potential to really help other people is really what I'm driving at.
absolutely.
And
I mean, that's something that we routinely see, as something that we strive for as a company as well, is find these links between disorders or other disease indications that may not have been uncovered before, because that's how you have the biggest impact in the most efficient way.
it's so interesting, and I'm sure we can talk again. I'd love to do that. From my side, just wanted to thank you again for spending the time with us today. I think you've got a heat wave over there in Boston, maybe at the moment. Somebody told me.
That's
But
right.
thank you very much for coming on and doing with us today. I really appreciate it. And just like to thank you so much for, doing what you've done, being so supportive throughout. And we're just really looking forward to the opportunity to do this type of work together.
Well, thanks so much, Chris. Thanks for the opportunity. It was really, really a pleasure to speak with you, and just, I think, together we can really change how we go about treating diseases.
Let's hope
Thank you.
so. And we're really, really, fingers crossed, going to make a difference for ARIA. And we're just really super stoked to have this opportunity to learn about it. We want other families to be aware of it. And, this patient-led space, or parent-led space is sort of, I suppose, increasingly a thing, and you don't want it to be a thing. And I suppose culturally, we found that in the States, people are more open to that than perhaps as is the case here in the UK. But we're just very grateful. And again, on a family level, everybody who knows Aria is super grateful that this is on the horizon for us. And so we'd love to, speak again sometime soon. But thanks for today. Appreciate it.
Thanks so much, Chris.